Hi. Now today we're gonna start discussing biomolecular principles. So understanding the central dogma of molecular biology and the concept of biological transcription and translation will be studied. As the first step, we want to start more delve into the DNA structure. So let's discuss genome, genes, and proteins. Genome is the complete set of genetic information which is embedded in our DNA. And what is gene? Gene is the functional unit of the genome. In fact, it's just physical segment of DNA that indirectly codes for a functional, the final product, which is a protein product. And what is protein? And protein is the molecular machines that's responsible for all the cells' structural and functional properties. For example, structural protein as a collagen, which is the most abundant proteins in our body, and enzymes are proteins, and antibodies, which will be an important immune function, as also protein. So in biology and biomedical science, the biggest question is, in terms of genetic point of view, how genotype or genetic composition determines our phenotype, which is observable traits such as eye color, blue eye color. That's responsible for development, physiology, behavior, and many diseases. So in the beginning, people propose the role of one gene will produce one enzyme or one protein. So it's one gene, one enzyme hypothesis. And that means a gene specifies a protein in a one-to-one relationship. While this is turned out to be not completely true, but this provides us a very good idea. So how many genes in our body? So the number of genes in the DNA, and that gene will encode for one functional protein. In fact, some genes encode proteins that are not enzymes, such as antibodies or structural component. And some genes encode a subunit of a protein, not a whole protein, such as a tertiary structure, which is a subunit part of a quaternary full functional protein. And sometimes some genes do not encode polypeptides, which is functional RNA molecules, rather than polypeptides, such as transfer RNA or ribosomal RNAs. So this is a reference material, but I want to go through a little bit of a detail of nucleotide, which is a molecular building blocks of DNA and RNA. So nucleotides are organic molecules that serve as a monomer unit to form the nucleic acid polymers, such as DNA and RNA. So these are real building blocks composed of 3 subunit molecules. So that is, you can see a nitrogenous base and 5 carbon sugar. You see this pentagon structure, and this is 5 carbon sugar, either ribose or deoxyribose. And you can see here, if this is 2, carbon number 2, if it's OH, that is ribose. And if there's no oxygen, that's a deoxyribose, then it becomes a DNA subunit. And at least one phosphate group. You can see here, 1, 2, 3 phosphate group. And so, except this phosphate group and these nucleotide called as nucleoside. And this is a nucleoside monophosphate, diphosphate, and triphosphate. And this base determines the kind of nucleotide. So you can see this a double ring structure, which are purines containing adenine or guanine, or single ring structure called the pyrimidines are cytosine, uracil for RNA, thymine for DNA. So the structure element of the nucleosides and the phosphate group bearing nucleotides. Now let's get into a little more detail of molecular basis of this DNA structure. And as I alluded, this 5-carbon structure of ribose and deoxyribose. You can compare which part is different. Yes, this number 2 carbons, and there's an OH, there's an H. So, and on the left-hand side, so this is adenine and thymine. You see there's a hydrogen bonding, 2 hydrogen bonding makes them as a complementary to interact with each other, while guanine and cytosine are having 3 hydrogen bonding. So that gives us specificity for AT or GC. In RNA case, instead of thymine, it goes to A uracil. And you look at this between thymine and uracil, what are the difference? You can see here, this is CH, this is CH3. So those are slight differences are between RNA and DNA. So DNA of a typical our human cell contains 3 billion nucleotides, which is amazingly huge, and which make up its genome, the whole nucleotide sequence. And gene is a segment of DNA that codes for 1 polypeptide chain. So that we call a gene. And depending on DNA and RNA, you can see what are the differences between here. We can see this 5 carbon sugars are different. So that's a pentose, either oxy or deoxy. And bases in DNAs are A, T, C, G, or adenine, thymine, cytosine, and guanine, while RNA, the others are the same, while there's no thymine, instead it's becoming a uracil, like here. And their physical properties could be very different. Usually RNA is present as a 1 strand. So RNA is known to be very unstable, while DNA is usually double-stranded, so it's more stable and sticky. So here comes a picture I put, and it's a historic picture, as this is James Watson and this is Francis Crick. And when they first came up with the idea of this double helical DNA model, so this is what they produced, a model of adenine, thymine, cytosine, guanine. So it's like 3-dimensional puzzle making. So puzzles are important, and the importance of this model is it automatically gives rise to the secret of heredity and DNA copying mechanism. Because we are always at the time curious how a single cell divided into 2 with exactly the same cell, having exactly the same genetic information. How would that be possible? And that's really important question to ask. And that functionality lies in the structure of this double helical structure. And right at the time, you remember that the Nature paper is 1953. So earlier than, Linus Pauling, who was a Nobel laureate and the founders of the field of quantum chemistry and molecular biology. And he proposed a nucleic acid structure, and you look at this. So here goes this nucleic acid structure. Several nucleotide residues are outside. So Watson and Crick, at the time they saw this, they right away recognized the problem of this, because this nucleoside residues are outside and sugar bases are inside. But what they considered at that time as a double helix model, it's just a beautiful thing because it automatically makes it the copying mechanism automatic, while this triple helix model doesn't fit. So they think this is wrong. And the influence of Linus Pauling can be lied in the Linux operating system, is that name is coming from the Linus Pauling. So let's talk about molecular basis of this genetics, which is coming from DNA structure as a double helix. So this dark and gray ones are a single stranded of DNA, which is supposed to be complementary each other. the important thing is there's a numbering here. That number comes from the numbering of 5 carbon sugar molecule. You see this adenine molecule, adenine with this pentose sugar, 1, 2, 3, 4, 5th carbon. So that from the 5th carbon to the next one connected is 1, 2, 3 prime carbon. So the connection from this one single strand is always 5th prime to 3 prime and keep going until 3 prime. So this we name it as a 5 prime end and this as a 3 prime end. So one is 5 prime end, the other is 3 prime end, and you realize the complementary structure will have to be the opposite way of this. So that's 5 prime end and this is 3 prime end to make this complementary hydrogen bonding. So this structure we call as a anti-parallel orientation. One strand from 5 prime phosphate here to 3 prime hydroxyl. You can see 3 prime hydroxyl. And complementary strand here is oriented in an anti-parallel fashion. So this formed DNA double helix, and the interaction between the different functional groups of DNA bases causes the 2 strands to adopt a double helical structure. And these 2 complementary strands of DNA align such that sugar phosphate backbone. So these sugars are outside, while inside these bases of each strand joined together. And that is the key idea of Watson and Crick base pairs. So, and let's consider a little bit how this DNA replication can happen. So their physical properties. So native state DNAs are usually double-stranded, and they hold together with hydrogen bond, which is about 1/20th strength of covalent bond. But there are so many hydrogen bonds, so it's like molecular Velcro, and it usually are very stable. But if he added heat or add OH minus, then they can interfere with the hydrogen bond and they can detach, and that we call as a denature, so that we can make this DNA as a single-stranded temporarily. And if you cool down, they actually come back to rigid. So that's we call renatured state. And this is very important to manipulate our DNA. So how to separate DNA reversible manner is by detaching this hydrogen bond, which can be denatured by heat. When the temperature is lower, the strands will renature or reassociate according to their ATCG base pairing rules. In fact, this forms a cool idea which led to Nobel Prize to how to amplify DNA. So a molecular biology technique to amplify particular DNA sequence, like exponentially, to study the function of DNA or identify a gene. This is very important technique called the PCR, or polymerase chain reaction. And that also is being used for detecting the coronavirus 19 for confirmation of the patient from their material. So next time we will discuss about in detail DNA replication and PCR further. Thank you for your attention.